Hello.
Morning.
Morning. How are you?
I am eating breakfast.
Okay.
It is earlier here.
Yeah, I know. Hello, Amanda.
Good morning. How are
Morning. All right. How are you?
you? Good.
Good. So we didn't have a meeting last week, but this week, we have some things to work on here. We have
It kills me every time you cancel.
a little bit inside, right?
But I totally understand.
Yeah. Looks like Lukas is here. Hello, Lukas.
Hi.
Hi, how are you?
So I'm just a bit sick, that's why.
Oh, yeah.
Yeah.
Welcome. All right, and there's Jesse. Hello, Jesse.
Morning.
Morning. So welcome. And we had two meetings this week. We had actually three. Well, we didn't have the DevoWorm meeting, but I did meet with Susan Crawford Young, who's one of the main regular people in the meeting. And we were talking a bit about some of her projects. She's working on a dissertation on like soft materials and biophysics of embryos. So it was an interesting discussion about what her advisors wanted her to do in the PhD is always a negotiation between your advisors and yourself on what dissertation or a document should look like that you're working on. So sometimes they want you to cover certain things, sometimes they want you to take it out, this and that. So, and she's been doing a lot of interesting work on that. So it's interesting stuff. And then I met with Morgan last Friday, and we talked about organoids and computational psychiatry. Yes. And then we this Wednesday we did the Cognition Futures Reading Group. We went through one of the papers that we had, just a little bit of, finished up one paper, the Voros paper, and then went on to the next paper. So I think that was pretty interesting. And then on Thursday we had, and it's Morgan and Amanda and Jesse and myself, a discussion on Michael Levin's work on regenerating brains. And so we looked at the squirrel, which has a lot of synaptogenesis associated with hibernation. There's the flatworm that you can regenerate large amounts of the body, and it retains its memories across regeneration events. And then other organisms as well. We talked a lot about that, and I gave some context on what was going on with the literature on that. There's some interesting older literature as well we referenced in there. So it was like, experiments they were doing back in the middle of the last century on things like the engram.
It was really interesting. There was like almost 200 references in the paper.
Yeah.
And I didn't get to all of them, obviously. I barely got through the paper, but it's really interesting in terms of it. That was a continuation. That was the final meeting of our in the series, basically, of it's sort of as sort of the Norbert Wiener cybernetics human use of human beings rating group, and we wanted to we finished that meeting last week, and I guess 2 weeks ago. And we wanted to kind of keep the momentum from that to take it into cybernetics a little bit differently, maybe, and with physical memory and all that stuff. And it was a really great meeting, and we're going to be on hiatus for that going forward for a little bit. But I think there's a lot of interest in pursuing things forward with sort of some things from cybernetics and work from Michael Levin and other stuff too. So I think it'll be a nice complement to our other stuff. And um, Amanda really has spearheaded the Human Use of Human Beings group. So kudos and thank you for that. And also this kind of picked up this other paper, and I think we have a lot of other things going forward too. Whether or not we go with Levin stuff ahead specifically or not, there was some really interesting stuff brewing there, and I'm glad Lukas can make it too, and other things. So hopefully, I've been thinking about it a lot, and I know it'll definitely be good. Part of just for other people to know, like part of the reason why we canceled it because we were doing a bi-weekly meeting, and with the holidays that are coming and everything else, it was just gonna we were gonna have like one meeting the rest of the year anyway, and it it, it was kind of like, okay, so this is a really good stopping point, but um, I'm really anticipating where we go next with it, and I'm already thinking about next year for that path. And yeah, I posted the meeting, and I posted Bradley's link to the meeting that Bradley recorded, thankfully.
Yeah.
So we'll see where that goes next. That was really great stuff. Yeah.
Yeah, I was happy to see, like what we've been able to do on that front. And we shouldn't neglect human use of human beings, at least in terms of like summarizing that and doing something with that. Those discussions, because there's a lot of things going forward in terms of cybernetics. So we were gone from human use of human beings reading group to cybernetics reading group. The implication being is that cybernetics reading group is kind of pulling forward stuff from human use of human beings and what are the lessons there, and then we're going to talk about some other papers and keep going like that. So we have some synthesis to do on that. But also, this is the last meeting of this year, so the next cybernetics meeting will be on like the first Thursday of the year, or the second Thursday, or something like that. We'll figure it out.
Yeah, and just a little context for anyone who has no idea about Norbert or human use of human beings. Norbert Wiener is sort of the cybernetics. Sometimes cited as the founding father of cybernetics itself. That it wasn't just him necessarily, but he wrote the book called Cybernetics and kind of gave it the name. Um, early like kind of proto systems theory stuff at Macy's conference where all these people are coming together to talk about this interdisciplinary stuff, and it kind of gave them that. But Human Use of Human Beings was this sort of essentially a public facing science book. I don't know if it's fair to say it's a public, science book. It is, but it was pretty like heavy and technical and steeped in literate. Like Norbert Wiener was raised by someone who really wanted him to be intellectually and academically oriented, and he often calls his father like the most greatest inspiration, and is also like his greatest antagonist academically and intellectually. Um, so he's like he's sort of a child prodigy who was manufactured to be a child prodigy, and I think is very sympathetic to a lot of things because of the different pressures he's been exposed to because of that. So you take that, and you take someone who's also like really gifted mathematically, and then this human use of human beings is sort of like this 1950s, 1940s era thinking and view about implications of where technology was going. And he had a really unique vantage point, I think, from being someone who was versed in a huge amount of backwards facing culture, as well as one of the few people in the world who kind of got what was going on generating information and the information systems at the time. There's an autobiography. No, there's a not an autobiography. The autobiographies are like talk like, oh, I'm a mathematician, and talked about his life. There's also something called like the dark hero of the information age, Morgan Hough, and it's sort of like um, it's just really interesting context. So I think it was a nice sort of historical piece and something sort of really almost painfully on point today, and like nothing has changed in 100 years almost. And some things were kind of a little whatever, but it we're kind of feeding it back into this reimagining cybernetics project, which has been kind of ever present, but always in the back of the room, not saying anything. Since I've been part of the lab, so I don't know, over 3 years.
Yeah.
And that's one of the first things I kind of worked on when I came to the lab, but we didn't really do a whole lot with it. So I think it's a nice mesh point of a lot of things, and also like some things related to conditioning futures and everything else, where we once again are really put on the face of where is discussion about the cybernetics topics, and what even going like back to the future in terms of like what kinds of measurements and understandings of relations about systems, and ho- ho- those sort of philosophical, but also like actually seeing the spaces where is the science of these particular, I don't know, properties of systems and change and feedback lacking. And I think we're looking forward to seeing how trying to do some work in those areas can be attached to quite a few other things. So that's exciting. That's kind of. I'm not sure I understand. Well, neither do we. That's kind of that's what we're trying to figure out.
Yeah.
So, yeah.
Siri got you. Yeah.
Thank you, Siri.
Well, thank you, Jesse, for that insight on the group. So, Morgan, you want to say something?
Well, I was just gonna add I mean, I totally loved everything Jesse was saying. His background is his relationship with his parents and what they were trying, and just also the context of the times. That particular period of time, and I was really lucky. My first mentor in brain science was Carl Pribram, who lived through those times. So it's like Carl Pribram knew Walter Freeman's father.
Okay.
Like,
the guy with the lobotomies, yeah.
So it was like active in brain science in the 40s, working as a brain surgeon in the 30s, but super interested in following the use of engineering and computers to aid that endeavor, and anyway, I just wanted to recommend Carl's autobiography, just because it's fascinating for the cast of characters that he's met along the way. He lived to almost 100.
Wow.
And so was looking back on where he was, anyway, and had lots of Norbert Wiener stories.
Well, that's great. Yeah, I've heard. Yeah, I know. I've heard of Carl Pribram, but I didn't know that he had a memoir. So no, that's great. Oh, he's gonna get it. Yeah. So yeah, I guess for the rest, I think we'll probably next move to maybe Jesse could pull up the Notion, because I saw in the Notion we have our papers that we want to review. Morgan has something to show.
Oh yeah, I'll get that ready. It needs to be updated back into the market.
Okay.
But yeah.
Oh, Morgan, did you want to?
Well, sorry, I was trying to. I thought I've got the autobiography right
Oh,
here.
okay.
I've been doing a massive reorganization of my library here, so it's been organized away.
Yeah.
I'll find it.
All right. Did Morgan? Did you want to present on something today, or were you?
I'm not quite there. I'd certainly love to talk about it, and I mean to talk about a number of things. I got quite caught up in the paper on the Judy Ford paper, although she's just a middle author, but cognition is entangled with metabolism.
Oh yeah.
And so I don't know if you want me to dive into this now or.
We could, yeah, we could do that, and then we'll do the
we can talk
board after that, or the notion after that. So you can go
okay,
first.
sure. I mean, just some thoughts on this. Really fascinating paper. It speaks to the kind of modeling that actually got me into computational biology or computational systems biology. In that. It was modeling. It was some work out of Yale. They were doing magnetic resonance spectroscopy. Yeah, thank you, Jesse. Yeah, that's the paper. Magnetic resonance spectroscopy to capture like functional magnetic resonance spectroscopy. In that, like capturing multiple time points to be able to track metabolism. So this is where you're using MRI like an NMR, right? And like with potentially with some spatial resolution, but certainly if you just focus in on a particular area, you're just grabbing time points, so you're getting spectra, right? So and usually when you're going to try and do this kind of, if you're going to do metabolic tracking, you're not going to use the endogenous metabolites. So NAA, creatine, choline, which are the abundant molecules that MRS is sensitive to, or that water's not blocking. But you're going to do some sort of carbon isotope and perhaps track that. Anyway, without getting into the details, combining something like functional MRS with EEG fMRI would be really interesting, and it was those kinds of models, just the simulations, that I started using Pisces, or Pisces and other tools like Copasi and Tellurium are all models that can take in SBML models. So this is systems biology markup language models, and turn the crank on the ODEs and can give you results that speak to the interplay of, say, in this case, I'm blanking on the guy. I'll check in just a minute. The kind of researcher at Yale who kind of led this work in the early 2000s, but you'd be looking at the astrocytes and their interplay with the neurons. So if you think of it as like dynamic causal models where you open up the number of cell types, right? And but very much the same kinds of equations that you're solving. It's just that now it's not just neuronal action, but also some additional metabolites, right? These are very similar. If you then go to something that, the kind of simple work that Bradley and I were talking about last week, just to get us started with like cell proliferation and differentiation modeling, looking at CompuCell 3D or Morpheus. Very similar. they also both tools will also take in SBML models and have all the math machinery to turn the crank on that. The difference being that those tools add the spatial component, the cellular POTS models, and or perhaps particle models that allow you to see some growth over space. So I haven't got there yet, but I know some SBML models that are available from biomodels that are very much like this Jacob et al. Paper that might be also good to just show people how to go to biomodels, download the SBML, open it up in Copacity or something like that, and run it and see, get some not necessarily results like this, but but you could compare different models that this wasn't so much a computational paper as kind of an overview, but really interesting and can be attacked with computational modeling like right now. Sorry, we've also got a bit of illness running through the household with small children. So really interesting paper, and I know Lukas had perhaps expressed some interest in were you going to work on something. This I think would be a really interesting intro project for someone to get into this kind of modeling, because again, not necessarily the spectroscopy data sets, but the EEG fMRI data sets are certainly available from like Open Neuro and places like that. So yeah, it would be interesting to again kind of take the DCM approach of modeling and just open it up and to tackle some of these questions that the Jacob et al. Paper raised.
Yeah, I think that's great. So yeah, Jesse put a copy of the metabolism and brain paper in the chat here. So this is Cognition is Entangled with Metabolism, Relevance for Resting State, EEG fMRI. So they're doing a combination of imaging modalities. There's Michael Jacob, Judith Ford, and Terrence Deacon, actually, too, who's on this. And they talk about sort of the entanglement or the contributions of metabolism with neural activity. So, we model neural activity as sort of like a neuron-centric view of neural activity. The neurons are generating action potentials, and then we might measure some more collective measure that, whether that or indirect measure of that, whether that be like a field potential or a some sort of hemodynamic response. But there are these other things in the brain, other cell types that are contributing as well. So this kind of goes into what's what they call flexible neurovascular coupling dynamics. So this is something that modifies cognition. And you can imagine, if you're if you're experiencing some sort of affect, like you're excited or you're angry, versus something like something where you have like high blood pressure or something. Those can all affect cognition. So in different ways. And so they talk about this, and they talk about neuropsychiatric disorders as like one application domain for this. So this is where something Jesse has been talking about with things having to do with neurodiversity and like whatever other kinds of things that we've been talking about lately.
I dropped in, both biomodels that again, if you're going to get into CompuCell 3D or Morpheus, so again, a lot of this started with just looking at metabolic networks and things that you'd see in typical biochemistry research. Okay, and so again, like, how do people figure out things like, you know, the Krebs cycle and things like that, right? That the metabolites all have a relationship to each other, and you can figure out the kind of network dynamics. This is something that again I was looking at back in you know, probably like 2005. So this is Aubert, but there is there's some Yale people who are actually collecting data that could be used. So I haven't fully connected this together, but like here's very much a model that you can take and you know, using capacity or I'm trying to, I mean, I think that's kind of like the most widely known one. Tellurium is a library that's come later, but they all take in these SBML models, and you're very much doing, you know, kind of advanced neurovascular coupling simulation here, because usually it's not including so much of metabolism. And you know, it's at least they, you know, again, coming from the MRI neuroimaging world, we're kind of more focused perhaps on, you know, capturing things like blood volume, and you know, fully separating out the Bold effect into its component oxygenation and blood volume changes, right?
Yeah.
Which don't necessarily relate, but bringing in metabolism, you know, is very easily done computationally with just starting with, say, this model here, and you know, like again, we can do a lot more with it.
Yeah, I think that's great.
And so, sorry, to Lucas's question there. Well, no, sorry, I didn't mean to say that they were similar. Both NMR is also done in the frequency domain. EEG might typically be, I mean, I'd say it needs to be looked at in joint time frequency. And people do joint time frequency of NMR too. Or like, again, like that's what you really want to do. The question is how fast you can capture that. So, and yeah, you'll see if you get into kind of advanced imaging of the you know, NMR MRI type. You'll then see about learn about hyperpolarized imaging, and these have to be captured very quickly. You kind of get one shot, but you get a really high signal. So it's it's the spectra is the frequency domain representation is the only similarity. You're looking at very different physics. And but I think it's really interesting to see, you know, like you know, Judy Ford, I can tell you, is from the Beagle lab. So I don't remember exactly what Beagle stands for, but it's something like the Brain Imaging and EEG lab. I think I did it justice. And so she's very interested in EEG in addition to MRI. And again, like EEG is giving you a direct neuronal measure, although you are getting it from a limited cell type or limited cell pool. You know, you're not getting electrical activity from all the brain. You're just getting it from pyramidal neurons that are oriented in a particular fashion, right? So that's why you want to couple it with these mathematical models that allow you to infer what the activity is in the other cell types, right? So I really think of it as you know, DCM is kind of a physiological EEG in that it's allowing you to get at kind of the hidden states of other cell types. And the NMR is giving you, in this case of magnetic resonance spectroscopy or functional MRS, you are also you are really tracking a metabolite. So you're perhaps using some sort of isotope of carbon in glucose or something like that to track its changes over time, that you can again focus on a particular biochemical model and infer time constants or things like that. So I hope that helps. But and happy to talk more about this offline, and for people interested in the kind of computational systems biology, tissue differentiation modeling.
Yeah, that's great. Thank you, Morgan. So we did have a discussion about like not just neuroimaging, but also like how to model an organoid, kind of from its origin point. So we said, well, and I actually did talk about some of this in a DVOR meeting. So it's on YouTube. It's several weeks ago, but we kind of we talked about this privately also in more detail. But basically, we want to model an organoid. We want to start with a stem cell. We want to proliferate those cells computationally. So we want to have a model that has like compartments or cells, and then those compartments, they proliferate in a space. So they proliferate into, say, a ball, and then those cells differentiate in some way. And so then the differentiation process in the cell is some model that we can specify, and then of course they have attributes of electrical activity or some sort of electrodynamics that we can simulate, but we have to have a good model internally of what that looks like. We also need a model of the cell in terms of how it differentiates. So that might be something like a genetic regulatory network or some other type of representation that allows you to have like a switch that says this cell is a stem cell, this cell is a precursor cell, and then it transitions to some cell type. So like, you have liver cells and brain cells and the different cells in the brain that we were talking about. So we talked about that part of the modeling of an organoid. And that's, kind of one of these Wild West sort of situations where you don't know what methods are best for that. You have to kind of test options for, how do we want to model it? It's some sort of hybrid model, obviously. Yeah.
It really speaks to what are your questions?
Right.
Yeah. It forces you to be specific about what you're hoping to get.
Right.
And then really tailoring the model, or hopefully finding somebody who's done something similar enough that you have a guide. Because there are so many different ways to do this work, and it really just speaks to the both the biological complexity, but also the needs to be really sure what you want to get at the end, right? So like, do you care the most about the 3D spatial structure, or do you care the most about the complexities of the internal biochemistry, for instance, or the biochemistry as it relates to the gene regulatory networks, right? So that interplay of proteins and the proteins that are involved in gene expression or suppression, right?
Yeah.
Like there's so many different aspects of this that have a remarkable complexity, right? So and I think we were talking a little bit about just the we can talk about neurotransmitter receptors, but of course the receptors themselves, I mean, how dynamic just the receptor density and spatial distribution are in fact on a cell, even though modeling wise we treat it as static.
Yeah. Yeah. So there's a lot of stuff to model, and one of the things about modeling is you want to put the things in that you think are most important, and it's an abstraction of like the real world. So you take the things that you think are important, you put them in the model, or maybe the things you think explain the most variance. So, like, with neural networks, we kind of use analogies of the brain, but we kind of take they've taken sort of the biophysical skeleton of what they think how communication works in the brain. And so the paper that we talked about with respect to entangling metabolism makes the point that, maybe that biophysical model of, the neural network or the perceptron is flawed. I mean, maybe we need to rethink that and add in other things, but we're doing this, of course, with data, and we're doing this in like with imaging modalities. But, you have this model basically of the brain. You want to just basically replicate how it works. And so you don't want to replicate every single thing. In fact, you can't, but you want to get the best sort of approximation of that. And so that's what we're trying to do. And we want to then be able to test it to see if it actually does something like the brain.
And just a final point to speak to, certainly Judy Ford's interest. I don't know Jacob or Deacon, but when they're talking about the clinical implications of this, some new work that I don't think I've posted yet in this form, but new work showing that patients with schizophrenia diagnosis who have elevated inflammation markers are responding well with anti-inflammation drugs. I mean, in their other, more psychiatric symptoms, and so that speaks to this, the physiology perhaps playing a role in certain subtypes or certain subgroups of patients, where it and again, this is kind of old hat in the depression research world. Ed Bullmore talks a lot about was one of the people kind of popularizing this in the inflamed mind, inflamed brain. His kind of 2010 book. Anyway, these would be these kind of biochemical biomodels would be really important in understanding how inflammation could play a role, for instance, in some of these things.
Yeah, thank you for that, Morgan. That was great. And yeah, Amanda said, awesome, thank you. So, okay.
I saw, just really quick to add to that, I saw a paper about psychedelics as anti-inflammatory agents. I think that was the exact title of the paper. So it would be interesting to see, like, there's all this hypothesizing about how psychedelics work, like what's the therapeutic mechanism, and it's probably, many levels. Certainly it seems like the psychological level for humans plays a role. Like there's something about the actual experience, but also there could, a big part of it could simply be like the physiological level, if they're like potent anti-inflammatory agents for brain cells specifically or something.
I mean, and so, sorry, one thing is that also Amanda and I met and talked this week, and which was great and super valuable. And we talked both about EEG modeling, but in particular, kind of physiological EEG modeling, or the kind of data sets that she has access to there are super interesting, relating to psychedelics, anesthesia, levels of consciousness, and so I don't want to. Let's at least bring up some of those papers, Amanda. But speaking to the kind of psychedelic modeling that I was talking about with Selena Adesoy and Morton Kringelbach, they haven't. It is not typical to have physiological models. They're very much doing, with their connectomic harmonics, they're still kind of doing um, either spectral modeling or spatial spectral modeling, right?
Mhm.
Which is, it's really interesting and it's important, but they're not tying that in a DCM kind of way to underlying physiology, and there were people doing it before Carl Friston, but DCM was like really the first implementation, kind of a major software package that everybody uses for analysis, which is why I use that. It's just easier to use that term so everybody knows what you're talking about. Lots of people were doing this work in the 90s before Carl even, touched EEG.
And so for everyone's benefit, what does DCM mean?
Dynamic cause, so it's the Phil's term, or Carl Friston and the Functional Imaging Lab in London's term for these kinds of ODE models that allow you to infer, allow you to estimate states of non-observed neurons or non-observed cell types, or get it micro parameters of the model, relating to time constants of receptors or and things like that. So it all depends on which model you're using. DCM popularized a particular 1995 Janssen-Ritt model, which again, if you go to biomodels or if you go to Model DB, which is kind of the same people who brought you Neuron C++ package, also have Model DB with lots of neuronal models. So Model DB focuses on neuronal models, and biomodels is from the European Bioinformatics Institute or something very generic like that. So they're doing plenty of non-neuronal, non-brain even modeling, as does CompuCell 3D and Morpheus, right? So the Morpheus stem cell 3D proliferation model is focused on intestinal cells. Dynamic causal modeling is very, very much, and there's two forms of that. One's for fMRI and one's for EEG. And the EEG one is the one that basically has the temporal resolution to give you a sense of the other cell types, whereas the fMRI one is very much kind of it's more of like a latent state model, and is not very much tied to the physiology. Or these are my opinions.
Yeah. Well, yeah, that's great.
I think other people's opinions too, but
yeah. They're not just half-formed. So yeah, that's great. Thank you. We talked about CompuCell 3D, and that's been something we've been thinking about in DevaWorm for several years. I actually went to visit the Center for Biocomplexity at Indiana University and gave a talk there once, and we talked about the models and how people were using them, and they're very flexible. It's a very flexible modeling package where you can build these what they call cellular POTS models. You have these small cells that are live in an array, and then you specify the parameters of the cells. So you specify the shape, the biophysics, the behavior of the cells, and then you can model things like sheets of cells, or you can model organs, other types of things. And they have, if you go to their community, which is actually quite active, they have tutorials. They have ways to apply different types of model systems to the model itself.
I mean, and he's got years of courses that he's taped and it makes available on YouTube. So it's not just CompuCell 3D that like all of their workshops and tutorials are online. You've got years of his network modeling, network dynamics courses, it's like a treasure trove of material.
Yeah.
Just amazing.
Yeah.
I was saying to Lukas that I like to build the source, supposedly for packaging for Fedora, but also just to get a sense of the package, and it's a lot like MATLAB in the sense that like it's really just a collection of numerical libraries, and it's like we here, looking at the dependencies are very similar to a bunch of other packages that are trying to model. It doesn't matter what domain you find this kind of common core of numerical and other geometric libraries that are used, and CompuCell 3D is very much like that. So that's where it gets its flexibility, right? That you're kind of just scripting together a bunch of other libraries.
So yeah, and so if people are interested in participating in this type of work, let us know. We could do it through DevaWorm, but we could also do it through the other, something else. So we're doing, we're interested again in maybe some of the neural work, neural modeling, and then also the organoids work.
And through Fedora and some organizations that are supported by like Red Hat and GitHub and related tech companies. I'm also trying to find computer science, computer engineering students that are interested in just learning software engineering.
Yeah.
And because again, building these packages, I find again looking across lots of these packages, there's a lot of similarities and a lot of you stumble on the same problems again and again. It's generally learning how to make CMake do what it's supposed to do. but getting into CMake and VTK, ITK, is a great. I think it's a great intro to a lot of engineering, work. So again, if there's anybody interested, you can go through Fedora and Outreachy for positions like that.
Okay. Yeah. Okay, thank you, Morgan. That was great. Great update. All right, I think we'll switch gears to Jesse. Okay, so Jesse here says, if you have a description or would like to solicit interns or something like that, let me know.
Yeah, NeuroFedora does that. Yeah, that'd be great.
Yeah, I mean, I'm interested in myself in getting into this space because I'm wading slowly into like actual data science stuff, but I'm still quite new and I want to get more experience in that. And it sounds like other people in the lab might be too, so I think there's warrants further discussion in some form. But yeah, let's. I don't know where people want to push that towards the DevaWorm or here or other things, but whatever it is, let me know or try to facilitate that.
I've certainly got more to say on the DevaWorm side, just because again, like Susan's doing a lot of finite element modeling, and that's always been my kind of real interest in electromagnetic neuroimaging. That's what we use, finite element modeling. So there's some great tools and some again kind of additional computational physics libraries that you end up seeing again and again and again that I think are of general interest, but again might come better from when you have a problem that needs it, and so she needs it because of the kind of I don't know how you characterize her work, mechanobiology of embryology, anyway, but I can give people good examples, for say TMS modeling, to transcranial magnetic stimulation modeling, or just high density EEG, source estimation, that use exactly the same libraries.
Oh, also, like, did Amanda or Lucas want to give any general updates? I forgot about that earlier.
I guess I'll say so. Next week, I'm not ready to share updates this week about the ISPSM talk, but next week I can.
Okay.
What's the ISPSM?
International Society for the Philosophy of the Science of Mind. It's a new group,
Cool.
and they
Oh,
have
okay.
an online conference in a few weeks.
It's a first time conference. It's like Ines Hippolito and some people in sort of the neurophenomenology, but phenomenology, I should say phenomenology, not neurophenomenology necessarily, and like other philosophy of mind stuff. We have a couple submissions there that I need to work on too. So it's just this endless thing of like it's coming, it's coming, it's coming, it's coming, and so on. Okay. So Bradley, you wanted to go over this list, or
Yeah, let's do that. So yeah, so I guess the Bika 2023 that's been closed out. That happened in October, middle of October. So it's been a couple weeks. I did an overview of the conference, and the presentation is done and is on our YouTube channel, and then the paper is coming out soon. Maybe the next week or so, it should be live. So the conference proceeding, they're publishing it. They get it out pretty quickly. So and was all kind of an ordeal on its own. So that was done.
I think Google Summer Code is completed too.
Yes, Google Summer Code is also completed. Yeah, thank you for everyone who participated. That was it was a lot longer than usual because they have these flexible project sizes, and we had an extension, and so I mean, yeah. So I think we did a good job there.
I don't know if this happened because I know there were certain blog things, but I'd really love to see like a blog recap from our participants because I feel like I know some of the stuff that has happened, and I know they've done a lot of work, but I would love to see like a final contextualization of it.
Yeah.
And I don't know what's gonna happen. Yeah. Okay. What else on this list here?
So we have Diva One Collaboration Journal. I think that's referring to the paper Lukas and I were working on along with some other people. That was we're going to resubmit that to the same journal. Not as well. We're going to resubmit it to the same journal. So that's still kind of, I guess, in preparation. So we don't have to like go into that right now, but just keep that on hold. And that I don't know what the schedule is for that because I've been trying to work on multiple things, and it's going to depend on me and getting that together. So I don't. We'll keep working on that. The Gibsonian information preprint. That's something that's still being worked out. I think the key here is that uh, we need to have like a primer of some type, something more detailed than because we had the conference paper that was actually for Bika, I think 2021, and then that was that I think that's gotten some buzz in the world, and so the next step there is to create kind of like a primer or something where we can have like more detailed things about Gibsonian information. So that's still in progress. That's going to be different from the conference paper. So I would just say for now, the preprint is in progress. Sort out. I think I think we've kind of sorted that out, but not entirely done yet, or to a point where it can be distributed. Anyway, so that's Gibsonian information. then the EI proceedings. So I submitted an EI proceedings. I think I talked about it 2 weeks ago, where we submitted another. We had this paper on continual embodied learning that we were doing, and that got rewritten as. So that was submitted to the EI conference proceedings, which was due at the beginning of this month. So that got submitted. That'll hopefully be, we'll see some movement on that in the new year, probably. The EI conference proceedings 2022, I still haven't gotten the final link for that. That's supposed to be in the process of being published. And there's nothing really that needs to be done on that. It's just a matter of getting the thing online. I don't know why it's taking so long, but suffice it to say, I hope it comes out in 2023. To be honest with you, I don't know.
Yeah.
But that's Jesse and myself, and actually, what's her name? Angela. Yeah. So that's, that'll be thing there. So that's EI conference. But then we should also add to this list, think about EI conference 2024. Think about what we might want to do for that. Like I've always said, it's good to think about your submissions in advance of what you're going to make before you make your move. So you have, I know we all have things that we want to do.
Accepting submissions now? Is that what they're doing?
No, not yet. But I mean, think about, just in the back of your mind, what would you like to do for that? What's viable to do, and what is, because not all of your ideas are viable for this. But yeah, I mean, and again, with the EI conference, it's virtual. You, I think, make one submission per person. So, it's kind of like when we did with NeuroMatch, where if we have some idea that we have a collaboration on multiple authors, one author submits an abstract for their thing, and then there's another person who submits an abstract for something else. So you can get a couple of submissions for the group. But I think we should talk. Well, when the submissions open up, we'll probably talk about it more as to what those ideas are. But I want people to think about it at least a little bit. And again, this is like embodied intelligence, so it could range from philosophy to robotics to modeling to whatever. But it's pretty diverse. They do tend to focus on soft robotics a bit, because I think the people who organize the conference are soft roboticists. So, but that's okay. I mean, yeah. So that's EI conference. I haven't heard anything about NeuroMatch. Yeah, I don't think that's happening this year. NeuroMatch conference. Hopefully, maybe they'll do something in the new year. But
I mean, is it just everybody's kind of going back to their regularly scheduled lives?
Maybe. I mean, it could be that. They have a foundation now, so it's like they have the summer school pretty fastidiously.
Right.
But yeah, I think it takes a lot of effort to organize it. So
it looks like it takes a lot of effort.
Oh yeah, definitely.
I mean, it's a good reason to get in touch with their CEO. Yeah,
Sorry, the screen's being crazy right now.
Okay.
Yeah, hold on a second. But you can keep talking.
Yeah.
Something.
Yeah. So I don't know what the schedule is for that, but yeah, I don't know what the.
Can I plug a Neurotech X thing?
Oh, go ahead. Yeah.
Yeah. So December 2nd, I think that weekend, is the Neurotech X Global Hackathon. If people are interested, they can check if they've got a local chapter, and if you don't have a local chapter, you can get in touch and we can help you make one. but I know, I'm seeing all the meetups announcements get postage for various cities. It is concentrated a lot in Europe, but again, it's global. So I know there'll be like active groups in India, as well as some in the States. I think mostly on the East Coast. But anyway, so check it out. Check Meetup, and or go to neurotechx.com. Yeah.
Yeah, that would be great. Yeah, I know that
So last year was really interesting. The brain age estimation from EEG, which again, is like remarkably, real interesting results that came out of that particular, hackathon.
What dates are those specifically?
So, like, I want to say it's December 2nd. Let me just double check on my calendar that I'm not. Yeah, so second, third, but it'll because it's starting in Europe. Like I prepare Americans for it starts Friday night.
Yeah. Yeah, it's always a problem with like being something happening on another continent.
Yeah. Well, so last year the French in particular had French and Spanish, Paris and Barcelona had computer science students involved. So there was they had already like about 100 students, and then there was a group in Serbia that also had maybe like 40 plus students, a lot of students, and so you like they were kind of dominating Discord, and during waking hours, but yeah, it's going on everywhere, or I think it'd be really interesting to see what the data challenge is this year, and if anybody's interested, just get in touch.
Yeah, that would be great. That'd be a good opportunity for people if you're interested in, getting into a project maybe that is, very collaborative. That would be a good opportunity. We had talked about doing this like last year. I know Ankit Grover was talking about it, and so we haven't really participated too much, but yeah. Oh, okay. Morgan wanted to say something. Yeah, so let's go back to the board and see. So, we have a bunch of things coming up. We're not really participating in them by presenting or anything, but they're opportunities online. So there's a Learning on Graphs conference as well, and that's happening around the same time. You can see on one of the lines there. That starts, I think, December 1st, and that's one of those conferences that they have like the virtual component. They have local meetups. It's hosted centrally, and so it's basically Learning on Graphs is all about graph neural networks. And the first conference standalone conference they had was last year, and I attended that, and it was actually very good. If you want to learn about graph neural networks, that's a good place to go to get some insights on what the state of the art is. We haven't been able to submit anything. I wanted to submit something on the work we were doing in DivaWorm last year and this year, but we haven't been able to really kind of get together a paper for that. So, but nevertheless, I think it's a good opportunity to learn. Some of these things are just kind of like, learning through osmosis. You attend a bunch of talks, you kind of digest them in a certain way, and then you kind of gain insights. We've in the past, what we've done is we've had a shared notes document in the lab. So if you attend a conference, you might have thoughts that you want to share with people, screenshots. You can put them in this document, and then other people can contribute. and you can have a nice like asynchronous back and forth. But that's something that we might do for learning on graphs or some of these other events. We have it looks like Jesse has a frontier map preprint that he's working on. I don't know what the status of that is.
It's basically on hold because of a bunch of other stuff.
Okay.
But it's still there. It's still alive. It's just buried under other priorities, including grad school and the other paper that's going on. So, yeah.
Okay.
Yeah.
Go ahead.
Sorry, my mics keep changing around, and also like I'm surrounded by this construction. I can't escape it. It's kind of crazy. If it's really noisy, just let me know.
Okay.
There's some other things I probably should mention, like there's I did not attend this, but there were like MIT Neurotech, like things I could put on here, but it kind of come to pass. Like there's been a bunch of local Boston events that I've been trying to sort out and attend when I can, but like the last 3 weeks I've had no midterm stays and everything else. I really haven't. I've been pretty like blocked away, relatively. But there's a bunch of stuff related. So let's move on to other things you wanted to put on the list, though.
Oh yeah. Well, there is another thing coming up, which is NeuroIPS, and that's usually an online event. I'm not sure what their sort of online component will be this year, but we always make a point of at least being aware of what's going on there.
The 23rd coming up next.
Well, it's in December. I think the beginning of December, so it'll be 2023. I'm not sure what the date is where it starts, but probably sometime in the first week of December. So that first week of December seems like that's pretty popular for yeah.
And at least for the past couple of years, Montreal's tried to do a NeuroAI thing that is associated with that, as well as one of the best things about that conference are the workshops associated with it, which vary from year to year, but there's typically like a medical imaging workshop. There's typically some sort of neural representations workshop. Always good to see what people put together, and saying the same thing to Amanda for the SFN technically started today and or last night, and there's usually pre conference workshops as associated with that too, that would be good to track. I know I'm supposed to be at George Washington right now. like actually, I think I've missed it. But like they did it in person only. For Neurodata Without Borders is like this morning DC time.
Yeah. Oh, wow.
And the Neuroscience Gateway, which is San Diego. Sorry, I was in the Neuroscience Gateway session this morning at which is kind of like a front end to the San Diego Supercomputing Center that makes all these neuro-related tools available via a really, really hard to use interface, which I really hope they get rid of. Together with the fact that they've got internet to links to all the big data repositories like OpenNeuro and all the data sets that Amazon hosts for Human Connectome Project, etc. So it's a great resource. I've pointed people to that don't have institutional high performance computing.
Yeah, that's great. um, yeah, so I think that's pretty much it for what we're doing now. And of course, we have the conference that's coming up. That's at the top. First annual web conference of the International Society for Philosophy.
Yeah.
Yeah.
There's also NAPWIC, which submissions are
Okay.
open. I'm help doing admin work and volunteering for them. I'm probably not going to try to spearhead something this year as like we always do these sort of like group things. I am probably not going to do that. If people are really interested in it, let me know and I can either co-sign it with you or just tell you how to submit to it. But NICWIC is the Celebration of Women Computing Conference. It's local. It's kind of a homegrown New York thing. It is open for submissions, especially for first-time researchers or whatever. I don't believe it's going to have a virtual component, but I could present vicariously for you if you really want to go and submit. You're seeing this, but yeah, that is also a thing, and I should mention that more in like the Slack and poke around with that. But that is also accepting submissions for about another month.
Yeah. All right. Yeah. So that's good. And so on this web conference, I is that like free attendance that's on the web, of course. We should make sure that people can register for that and everything.
The ISPSM one?
Yeah.
It is free, I believe. And it's pretty, it's very, very first time, but I think it's, I think they're trying to make it as accessible as possible.
Yeah. And so for that, if you have recorded your talk, please let me know. I can put it on the YouTube channel so people can see it. But if I guess they'll be recording the talks too on their own thing. So we can do a number of things for that. Maybe we'll also promote some of the our stuff for that, and so that'd be good. So I think that's all for now. Thank you, Jesse, for going through that list. If we have ideas, please put them in the Slack or bring them to our attention. We can add them to the list. If you see opportunities where we can submit things, especially, please have us put them on the list so we can target them. So
something you potentially put on the bottom of the list towards the bottom, because they're sort of like our spring updates, one of the things people doing at the admin level. Just to say this and just to kind of nudge stuff, like if open for those who aren't really familiar like with a lab, like how do you contribute or do things here? Like you can say you want to lead a project. Like we have a lot of opportunity for project leadership or for even like collaborative leadership if you don't want to do something yourself. Like if you want to start something, if you're looking to start something, you found the right place, buddy. And that's here in Orthogonal Lab. But what I mean by that is more like, if you like, this is kind of something I'm working with JoPro, like why I made JoPro too. Like in a sense of like Bradley's always offered this explicitly. Like Bradley is really great at like, hey, let's help develop something for you. So like I'm not trying to say, oh, this is a JoPro thing, but like there's a lot of opportunity to get like some training to develop projects. And that's one thing, but a related thing is like I haven't done anything concretely yet, but like we're at the season of time to start thinking about, I don't know, spring semester or January, February, March time, because we can create solicitations for a variety of things. One of the things that really coming up is like, I think we might need like a sort of like science communicator or like assistant lab manager or maybe just a different lab manager kind of a thing to do some of the like recapping our meetings, maybe clipping some things from YouTube, helping with some of designing our other stuff. Like there's a lot of opportunity there for that, which is, great from an admin and like marketing putting out there standpoint. But also like you get a lot of experience with how do you do, what's it like making all of this stuff happen. But even outside of that, like there's stuff in cybernetics. I'm going to be doing some things for like philosophy and history of science specifically that I'd really love to have an intern for. There's some things in maybe kind of like I wanted something in the space of the Logiciel book with the Johann talk, like kind of some edges of mathematics thing. It kind of might tie into some of the cybernetics thing, the reimagining cybernetics project we've talked about. There's a bunch of these projects that are kind of on the horizon that we're looking for both leadership and also just like we're going to be spending some time in the next month or two crafting the more of the project and also trying to find ways to solicit like basically advertising for interns at that level, but also like a reminder, like it's not just, you don't just have to be an undergraduate student who's looking for some like potential academic credit or internship. Like if you want to lead projects, and we kind of have some of that developing here, you have opportunity to do that too. So that's my advertisement for that side of things.
Okay. Yeah, it's great. Thank you, Jesse, for that. Now, I guess Morgan put some things in the chat, and I'm going to share my screen again. Oh, go ahead.
I put some things in Slack too. Just sorry, go ahead.
Yeah. Oh, okay. I'll just go through the links here.
Sure.
This is a mixture of large-scale dynamic functional brain network modes.
Yeah. Yes. This is some new work by last author Mark Woolrich is now the head of the MEG Center in Oxford. So is very much coming out of FIMRIB and the variational Bayes modeling of fMRI, and but I know that he's been focused on electrophys now for over a decade, and this is really bringing this dynamo modeling is really adding a next generation analysis to electrophys. It's really I think a culmination of a lot of his work on this, just wanted to say that this, if you get into it, it requires a little bit of background reading to see some of the in Markov model stuff that he's done previously, microstate EEG analysis as well. So the way that they are kind of automating a temporal segmentation of the data stream. But again, super interesting. Always you're always going to learn from Mark. Yeah, and so it's time well spent. And this was, I think, the final kind of recommendation that, or the final kind of goal that we set, looking at the kind of data that this high, what I'm going to call high density rodent EEG with physiological, with drug challenge, that I think would be really, really interesting. And again, this is tools that are available from their GitHub, and just wanted to talk about this and talk about the biocybernetics lab in terms of learning from graphs.
Yeah.
his work, which goes back to Stefano, goes back a long way, but again, it's kind of taking these biochemical mathematical models that have been used previously, but he speaks very, very clearly on the mathematical issues of system identification and their confounds. So there's some really interesting math problems to doing, well, as well as the fact that he literally calls himself a biocybernetics lab, right?
Right.
So I thought, again, like I like people who are using these terms and are generating computational tools for them, right? And he's doing that. If you see these papers, you can see some of the math issues that are coming up with this, and a tool, Pyrates, which I think is similar, but is a more more recent. Let me just get their particular. This is Max Planck, Leipzig. So let me just figure out. Again, just pointing out tools that people can, use and play with now to get at these particular to find projects that you can get involved with now, and software that you can be using now. So Pyrates, again, coming from an old school EEG modeling lab, but these are some students of his that have generated a tool like OSL Dynamics, the Dynamo tool from Woolrich. This is using TensorFlow on the back end for some things, but most importantly, it's got these kind of explicit models that you're using to get at unobserved physiology, which I'm going to keep banging on. Want to make neuroimaging physiological, for it to be useful.
Yeah, thanks for the resources.
And then in Slack, I just in addition to the NeuroPS or NeuroPS workshops, they're also doing a bunch of tutorials too, so people can check those out. And hopefully, I'm seeing more and more conferences going back to the old ways of in person.
Yeah.
I hope they don't. I put it events. And I should have dropped that in random. Wikipedia is doing celebration here. Wikipedia deserves more celebrating.
Oh, definitely, yeah.
But the link below is for the tutorials.
Okay. Oh, that's a NeuroIP. Oh, that's just from their blog, NeuroIPS. They're involved in NeuroIPS, or? Oh, you're muted. I don't know what happened to the audio.
Okay. Machine learning for theorem proving, application development using large language models. There's some really interesting tutorials this year associated with the conference.
Yeah. Okay, these are the tutorials for NeuroIPS.
Yeah.
So this actually has all the tutorials. Maybe we'll do this next week, going through these and see. Yeah, one thing I wanted to mention about which one of these was it, where they were talking about, it was the paper where they were talking about they were talking about neural models versus, or the dynamic causal models versus hidden Markov models. So it's not it. This one here. So this is a mixture of large-scale dynamic functional brain network modes. This is where they have this model Dynamo, which is dynamic network modes. And so this is actually, when you said, you start talking about hidden states and things like that, I thought of a hidden Markov model. But they're actually improving upon the hidden Markov model in terms of what's going on in the brain. So this is where, Dynamo is kind of like an improvement on just taking hidden Markov models, which are used in a lot of different application domains, and they're bringing it into the neuroimaging space, but they're improving upon that so that you can find things, hidden sources of explanation, hidden variance that explains things in your model, and they're using this. So this is interesting how they kind of break this down in terms of how it improves upon the hidden Markov model.
Yeah. And you'll see, and again, like if you're following the research in this area, applying these recurrent neural network models have become a kind of subtopic in itself. So there's a lot of interesting literature and some groups that are doing some like there's a psych RNN project out of Yale or Brown, but again, like the same people who gave us the hierarchical diffusion drift models for modeling reaction times together with EEG fMRI. They're now applying these recurrent neural network models. And again, like Mark Woolrich is going to always be someone you're going to learn from. So this is again, like I feel like someone who's taken all of these different threads together and tried to put together, a comprehensive end-to-end tool, as is typical for the Oxford group.
Well, thank you, Morgan. That was great. Great overview of that. I have a couple of links too that Morgan had shared in, I think it was computational psychiatry channel. So this is this tool, Centile Brain or Centile Brain. This is something that it looks like it's a data, like it offers you data sets from the Enigma lifespan working group. So this is kind of like developmental data.
So I can just say a little bit about Enigma, right? So Enigma, which I'm a part of through my old PI at Oxford, contributes. Everybody, all those psychiatry groups that do neuroimaging, contributes to the Enigma pool of data, right? And this is so because of that, they've got like large data sets of common diagnoses, as well as small data sets of less common. And there's an EEG working group, there's a PET working group, all sorts, right? And then perhaps also people who are like looking across all this data, where it's like we've got kids who've got first episode psychosis, or adults who were given a diagnosis as a young child or something like that, right? And again, it kind of speaks to the wonderful results that we got at the Neurotech hackathon last year. Just using EEG data that was derived from the Child Mind Institute Healthy Brain Network, which if anybody's seen me speak on YouTube before, I go on about incessantly. It is an incredible resource, underutilized resource. So yeah, it's been hot to use MRI to do the same thing, right? Is to look at trajectories and and the reverse of that is where does some particular brain sit on that model, in terms of is there a mismatch of biological age from their estimated, but it's an incredible group that is managed out of Paul Thompson manages out of USC.
Right. Yeah, so that would be interesting to get into, a little bit if we have the interest in not only in development but also in critical periods more specifically. So this would be, maybe find some data to match our models. And then this protege, which is another, this is developed by the Stanford Center for Biomedical Informatics. This is where they're developing cutting edge ways to acquire, represent, and process information about human health. So this is another set of. Do you have anything to say about this, Morgan?
Just how difficult it is to capture all the different areas here, right? This is a completely different kind of tool.
Yeah.
But still so important, right?
Yeah.
So for kind of collating and for organizing the just wealth of biological data that we're going to be continually inundated with over the next decades. Right. That more and more it's become necessary to use machine learning to do the organizing, right, and to find the patterns or to find the connections, and the ontologies are kind of the informatics tools or frameworks for allowing that or giving that some structure, right? Yeah. So this was just one example. Again, I can't say I follow this particular world very well, but I wanted to at least flag it as another thing to watch in terms of especially biological data management.
Yeah, looks like they're developing ontologies, which are these sort of descriptive languages that kind of sit on top of the data, and it's a way to classify the data so you can have like a framework for understanding what the data is and what the variables mean and things like that. So this is nice to have these kind of tools because they're just sometimes as important as making data available. So I wanted to go over those because I thought those were interesting as well. So it looks like Lukas had to leave, so thank you for attending, Lukas. Now I'd like to move on to some other things. Let's see. We have a couple things, announcements. So first of all, as Jesse mentioned, NICWIC 2024, the call for proposals is open for submission. That's due on September 8th, so this is submitting an abstract for a poster or a talk or a panel. So last year, or in the last couple years, Jesse has hosted a smorgasbord of different topics. So we'd have like 20 minutes, and we'd have these different topics that people would contribute maybe 5 or 6 minutes to. And so I guess Jesse doesn't have time for that this year, but we might have someone else in the lab take up the mantle instead, or I think this is all live. I don't know where they're hosting it this year. Contact Jesse if you're interested. If you're in New York, I guess, we'll see how that goes. But that's coming up, and that's something we participated in for the last few years. This is actually something that I found. This is a little bit different from so in the Cognition Futures Reading Group, we've been talking about the brain is a blank. And this is Jesse is working on this presentation for IPS PM or whatever it is, that conference that we just talked about. And this is, the idea is that the brain has been given a lot of metaphors. So, there's the brain is a computer, the brain is a orchestra, the brain, you can think of a number of different metaphors. And Jesse's been working on a nice set of, like a nice historical overview and sort of survey of some of these metaphors. We also talked about a paper that was about some of these metaphors. I think this was in another Saturday meeting. And so, that they're metaphors at the metaphoric level. Certainly, there's been a lot of thinking about that, but this is actually an interesting sort of data-driven characterization of the brain. So, this is the brain is ... And instead of inserting a metaphor here, like a computer or something like that, we're going to insert like different biological terms or maybe cybernetic terms. And we're going to look at, and I don't know if this is like from the literature or what, but this is actually, I think maybe not, not at the level of a metaphor, but at the level of what the sort of the study was about. So I guess it is sort of about metaphors, but also about some of these more biological terms. In any case, they do a dimensionality reduction of this data set, and they basically have these different descriptors. So there's the brain is a cholesterol-rich organ. The brain is an energy-demanding organ. So there are these physiological definitions. A lipid-rich organ. And what they're doing is they're putting it in this UMAP space, which is just basically like if they cluster together, they're similar. And then there's some distance metric, although with UMAP, it's never clear what that distance metric is. But basically, we have these different categories of thing that people have used to describe the brain. And some of it's metaphoric and some of it's not. So like an information processor is metaphoric. An integrative, interconnected system can be both metaphoric, and you can do studies like network science, or you can do different types of like modeling. You have a complex network, which of course we do things with brain networks and connectomes, and there's a formal methodology for that. Along with that is a complex dynamics. Could be dynamical system, but they say a complex dynamic system. They have a picture of an attractor base in there, so you have this, this mathematical characterization of the dynamics. A nonlinear system, a heterogeneous organ, meaning that it has multiple cell types and things like that, as we were talking about. An immune privileged organ, a highly vascularized organ, a common metastatic site. So that's very specific to cancers, brain cancers. So you can see that you have these physiological definitions, very purely physiological. And, it's like the brain is probably all of these things in terms of the physiology, but people focused on one thing or another thing to sort of name it. So, you have the organ of stress adaptation. Well, that's just the topic of the paper. They were exploring it in the brain and that sort of thing. A plastic adaptive organ, so it's a descriptor. A control center, which is another metaphor, because, the control center being like the function, but also like what would you describe it as? So, this is a nice graphic. I can't actually remember where I got this from, but it just shows, I thought this might be of interest to Jesse because he's been working on these metaphors sort of historically and sort of, in a philosophy sense, but this is also another way. I think this is based on literature mining, so this is a little bit different approach, but still, people are thinking about the brain in different ways, and it's interesting to see how those fall out in some sort of space, even if it's hard to interpret.
But just how important it is to really capture all of those different facets, and like it's very easy because of the difficulty to even follow one thread of those.
Right.
You can lose yourself in one thread or one facet,
right.
But as the Deacon Ford paper, sorry, I forget the first author, but the metabolism could play a cognitive role, or it could have a cognitive influence. So I mean, it's that first link on the. Sorry, I can't see it now, but the first link about not cholesterol, but
cholesterol-rich organ.
Yeah,
energy.
sorry, cholesterol rich. Yeah, yeah. I mean, something as simple as that. It has a fascinating thread.
Yeah.
That speaks to perhaps myelination and connectomics.
Right.
Like without it, and again, like it'd be so easy to ignore it. Uh, but all of these are going to play together and have some influence. So it really is like you got to find out where that came from.
Yeah.
Yeah. And
Oh, go ahead.
go ahead, Bradley.
Oh, I was just going to say, this ties in very nicely with the shallow brain hypothesis we talked about 2 weeks ago, where you have the neocortex, of course, that controls cognition, but you have these other areas and it kind of spreads out from there. And the same holds true for some of these other factors like, physiology and other things that feed into the brain. So that's another point. Jesse, you were going to say something.
Yeah, I was basically going to say, this weird noises behind me continue. I was going to say, it's just kind of a plus one to what Morgan said, and it's sort of like one of the things I would love to really articulate well in the presentation, the papers that I'm working on is, it's the easiest thing to do is to talk about one really, particularly if it's accessible function. And that's sort of like to dive into like deep phenomenological stuff, like there's reasons why that's the case from like a human or a biological agent perspective. Like we can isolate this, okay, here's a causal, strand or correlation strand and a mechanism, and we can poke this around and it dives way into the philosophy of science stuff too that we've been talking about. But it's like, yeah, we're still trying to find good ways to even conceptualize, like, something that could be all of those things listed out. And it's hard, like, how do you keep track of the strands? And what is that? What kind of, like, pressures, like, even, like, epistemological, ontological, like, all these different ways of trying to, like, how do you, like, it's, there's such a convention of, okay, well, we're going to look at this thing, and you kind of have to, like, when you're writing the paper, you have to talk about one topic. You have to say, this is it. And it's just like understanding there's a lot there. And so in the presentation that I'm working on, like, I don't know how great I'm going to do it, but like, I really would like to kind of get at that point. And then also there's sort of the over time thing, like there's sort of a requisite, I'm kind of looking at like the cultural, like affordance, like the zeitgeist of like what was available and how does that affect a common understanding, like you could say it's, you can say part of why you can say it's a computer now, why that analogy is so great, is that we have computers now and they're much more available now. Obviously, cognitive science kind of came around through this computer analogy era when computation was starting to really have its birth, I suppose. And then all the summers and winters and stuff, but like just things like that. And I don't want to ramble, but like it's a really interesting point and it's a really hard point. It's not just the paper that Bradley was showing in the chart. It does really stress that it's not even just it's not even a one-dimensional thing to talk about. You're kind of entering inherently this really complicated space. And yeah, I'll leave it at that.
Yeah. And so thank you, Jesse.
And just to speak, like at a funding, like the GRASP lab is the robotics lab at University of Michigan.
Yeah.
And the speaker this week was talking about, was working on kind of exoskeletons for ASL, but was, at the Q&A period was talking about the difficulty of, was like NSF. NSF doesn't care about producing tools for patient care. And NIH doesn't want me working on engineering. Doesn't want me building stuff. And so it comes down to a funding, or there's some institutional issues that come about too. That I thought he articulated well.
Yeah. This is a bioarchive paper that Morgan shared. This is the OSL dynamics. This is a toolbox for modeling fast dynamic brain activity. This is similar to what we were talking about before with some of these tools and packages and doing some of these analyses.
Yeah. And he's literally naming it the same way that FSL was named. It's just that, his institutions begins with an O, so it's OSL.
Yeah. Great. Thank you. Jesse posted this a couple weeks ago. This is kind of switching gears a little bit to like project management and some open source stuff, but this is something, this is a teaching and learning paper. It's a conference paper on live coding, a review of the literature. So this is something I actually have been getting experience with this semester in terms of teaching via live coding. And doing demos in front of the class is very much a demo or die aspect that we've talked about again and again. And this is about, having a class where you go in and you present, you basically code in front of the students. Actually, I don't really do it that way. I like to just bring in examples and then walk through them and then maybe run some variations on it. So, if you have a piece of code, and I usually use like a Colab notebook or a Jupyter notebook to do this. You'll have some code in the notebook. You'll have a kernel, and then you'll kind of walk through the kernel. You'll have comments in the kernel, and then you'll maybe change some of the parameters and run it in front of the students. And you can structure your whole lecture around that and say, here's an example of this type of thing that we're talking about in class, like a for loop or, a classification model. And then, kind of walking through the code, what each line does, and then, kind of run it with maybe some variations. So, you might show where it breaks. You might show where it shows a different output. And so those are very effective teaching tools. And I found that, it's definitely something that, if you're teaching coding, you basically have to do it like that. You can't have like a PowerPoint presentation and say, here's the code, just kind of commit it to memory, or, you want to have a more interactive method for that. So this is nice. This is about computing education research, this paper. They basically, in this paper, they did like they looked at the literature on this in computing education research. They investigated the value of live coding in an educational setting. They had different papers in different categories. So, one was about how different papers define live coding. So there are different ways you can do this. 2 is whether its version of live coding could be considered active learning. So is it active learning? Now, different people have different opinions on this. This is why it's kind of, that's why they're doing this whole analysis. They're kind of trying to get at like what it is about live coding, that varies across people doing this in practice, and then what's the most effective method. There's also people have used different types of data analysis. Then there's evidence provided for the effectiveness of live coding. I have to say that, like, based on my own experience, which is very limited, it does seem like there are a lot of subtleties to live coding that make it effective. Like, you can't just sit there and write a bunch of code in front of people and not explain what you're doing. It's really about walking through a problem. It's almost like teaching mathematics, where you have, like, you write on the board, you write some equations on the board, you walk through the equations, Maybe a little bit more interactive than that, though, because you want to explain, especially if it's an introductory course, what each line of code is doing, and you want to repeat yourself, and then you want to say, okay, what if I change this parameter here, this variable? What if I change this line of code? And then show when it breaks, and show when it's a successful run. Those sorts of things. So this is all like there are different ways of doing live coding. Um, Reported benefits and drawbacks of live coding, theoretical frameworks used to explain the basis, effects, or goals of live coding. And then we found that although live coding has been recommended as one of the best practices for teaching programming, there is a lack of empirical evidence to support claims about the effectiveness of live coding on student learning. So this is interesting that, this is like kind of a common assumption. I know that in the like in the carpentries, which is a group that teaches coding to people, it's kind of in an introductory level. They stress live coding quite a bit, and of course, the class I'm teaching, it has a history of like where people you use live coding to get the ideas across. But this is something that there isn't a lot of hard evidence that it actually does improve student learning beyond like just them reading about basically either you give them an exercise kind of on paper like in a book, or if they practice on their own. So this is something that is a teaching tool. I found it very interesting because it really does kind of it's like any kind of teaching technique. What is the value of it? How can you improve your efficacy at it? And there are different things you have to consider. It's not just like getting up there and start coding and showing people things. You have to really kind of put some effort into making it effective for people. So, live coding can demonstrate well accepted programming practices like incremental coding, debugging, testing, and commenting. So you can show all those to people how they work by doing them, but also by interacting with the code more than you would if it were just in a textbook. Yeah, so this is nice. This is a nice paper. Thank you, Jesse, for finding this. Definitely, there are different, you can even have student-led live coding or collaborative live coding. Some of these, however, in the class are hard to implement just because of the ed tech that's available. So if we're in an environment like this, oftentimes in our meetings, we've done, especially in DevoWorm, we'll have live coding sessions. Actually, our GSOC related work, we always have live coding demos where we have people doing a demo of their work in a notebook, but they don't, it's not interactive. I can't touch it. We just someone sharing their screen and they're walking through their code a little bit. We usually don't go deeply into the code though, and it's just because of the constraints of our meeting space. We can't we don't want to dig into the weeds of it. Oftentimes it's not necessary, but oftentimes it's also kind of technically onerous to do that. So I really I think there are different ways to do this, and maybe as we continue thinking about our open source meetings, which we've taken a break from for a while, maybe doing more live coding in those kind of meetings would be useful. But, implementing it is going to be a little bit tricky. So I think this is a nice overview of some of the practices and how they vary. Definitely something to follow up on. Okay. I don't know if Jesse wanted to say anything about that or.
Nothing in particular, but I'm like interested in. I'm hoping to kind of follow up with some of the professors have mentioned it. Or one of them was on the paper.
Oh yeah.
Because I'm kind of I'm with plot twisters doing some ed tech related stuff. I'm interested in like education, and pedagogy, and technology in general. So if people are interested in that, reach out to me because there's things happening maybe into next year on that too. But that's all. It's interesting space.
Yeah, it is. So the last thing I wanted to talk about today was this paper. As you see, I kind of came prepared, but we. So this is a new paper. It came out, and we've been talking about in our Cognition Futures reading group, or actually in this group too. We've been talking about the CBMM group at MIT and how they've kind of their take on AI and how they think NeuroAI is kind of played out. And
well, just to be clear, that was a invited research people speaking at CBMM. I don't want to mischaracterize CBM itself as having that viewpoint, because people there are it felt honestly like for example, Pojo, who was kind of chairing everything, was actually quite sympathetic to NeuroAI. And I don't just to say, like, at an institutional level at MIT, I don't think CBMM is trying to dump NeuroAI off the table.
Yeah.
However, big names in AI research have, at that event, have very much kind of made NeuroAI almost seem outdated. Like if you were a computer science person looking at AI at that event, and many of them showed up only to that panel, and you heard that from like Demis and the other, like, some other people at, like, the higher end of things, like I think Jeffrey Hinder was a part of it too on this thing, right? You'd think, oh geez, well, like, yeah, I'm not going to be stupid. I don't even want to talk about neuro AI at all. I just want to focus on this other stuff. Like, I think that there was a valid thing. I need to write up something about this because we keep talking about it, and I want to do it justice. But yes, that's my disclaimer.
Yeah.
I mean, there was that just crushing Twitter comment from David. David Fitz. It's like spelled with like a PH. Like, sorry, I'm blanking on him, but like he had something like, you guys are you're just so behind. Like, we're at DeepMind, like crushing it, with real results, and and you're talking about like we should look at the brain or something like that.
Oh, okay.
The difference being, so again, as someone who is interested in NeuroAI for psychiatry, right? like not interested in machine learning for its own purposes, or for its as a separate research area, right? This is like NeuroAI is about better simulations,
right?
Okay, for me, or like again, like what Bradley and I are talking about is like making computational psychiatry more computational biology, right? Yeah, and so like again, like I think it's super important to like, what are people's goals? And so like what, because I think there's a bunch of people in NeuroAI, sorry, that's a puppy, is not being tortured. He just wants to go. She just wants to go. I'm looking at this list of authors, and like there's so many different people, so many different interests here. I mean, I just posted a recent Bruno Olshausen paper, which was like super math theoretical in its focus. Terry Stanowski, obviously, again, like there's lots of people, there's so many different varied interests in these, and it's just I think it can be a really difficult term to get everybody to agree what the hell they're even talking about.
Yeah, I know. It's so that's what where this paper comes in. So the term NeuroAI and this paper.
I'm gonna get the puppy, sorry.
Okay, that's okay. So this paper kind of is a position piece, which is always kind of provocative in and of itself, so just keep that in mind. This is catalyzing next generation artificial intelligence through NeuroAI. So this is NeuroAI, which is, of course, using the brain as inspiration for AI. There's certain schools of thought on that, but it's basically, approaching AI as an enterprise of looking at the brain as that analogy or is that set of mechanisms that we want to model. So we have a bunch of people here, a lot of them from are famous, a lot of them from the Neuromatch arena. Anthony Zador, Blake Richards, Yashua Bengio, Albana Bolahin, Matthew Botvinnik, Ann Churchland, Surya Ganguly. So these people have different backgrounds and different things.
Yeah. I mean, one's a neuroscientist, one's a physicist. Just take your last two examples, right?
Yeah, definitely. So there's a wide range of sort of opinion here. People working on different problems. Adam Marblestone being like more of a computational biologist, and that background. So this is a nice author list. And so their position is that NeuroAI, or at least the way they're basically describing NeuroAI in this paper, is the path forward. So the abstract reads, neuroscience has long been an essential driver of progress in artificial intelligence. We propose that to accelerate progress in AI, we must invest in fundamental research in NeuroAI. So this is where they're just basically, it's a call for funding and more research. This is the way forward. This is our position. A core component of this is the embodied Turing test. So this is interesting. This is actually something that they're going to talk about in the paper, which challenges AI animal models to interact with the sensory motor world at skill levels akin to their living counterparts. So this is where you have some sort of AI animal model. Like, we could talk about C. elegans as a model where we have the connectome of C. elegans. We know what it does to a large extent. We also know what the body of C. elegans is. In OpenWorm, we do these biophysical models of the body, but we also have these models of the connectome, and then it's connecting those together, but also then having this Turing test, which is like an embodied Turing test, which is different than the Turing test that we've talked about in the past. But basically, it's a way to detect whether something is living or not. There was a thing that people have done this in the A-Life community, and there's been some talk about this in OpenWorm as well, where like if you take an image of like a behaving animal and you compare it to maybe like a simulation, can you distinguish between the two? And this is something, actually, if you've ever seen the show Devs, which was a show on FX, I believe, they had a scene in the first episode where they were doing this. They were at this fictional computer company, and they were trying to simulate consciousness. And they had these programmers. It was kind of, I always thought it was kind of like inspired by OpenWorm, but where they had this programmer who was simulating a C. elegans doing like a omega turn, and they have this thing where they show the simulation mimicking what the organism is doing. So that, I think that's what they kind of mean by embodied Turing test. But anyways, I mean, there are a lot of issues with that in terms of like, can you replicate the behavior? Is it the same thing as what's going on in the biology? Because if you have a simple enough set of behavioral repertoires, you can simulate those pretty easily using physics. It has nothing to do with the brain of the organism. So that's one like benchmark we could use, but I can see right away that maybe, based on our own experience, I think maybe we need to go beyond that somewhat. In any case, the embodied Turing test shifts the focus from those capabilities like game playing and language. So this is something, again, like, the traditional view of AI has been to do strategic things, game playing, language. We've done video games, we've done games like Go, chess. And then we've also done language tests, so we have large language models. This, of course, this embodied Turing test goes beyond that, and it's in a totally different domain that can be defined by the animal model. So, this is something that we need new tests if we're going to say model fruit fly and their behavior. And so this is something that we need to think differently on. So this focuses from different capabilities that are normally associated with cognition that are especially well developed or uniquely human to those capabilities, inherited from over 500 million years of evolution that are shared with all animals. I would point out that this is maybe not the best way to phrase this. That, first of all, we don't necessarily care if it's human behavior. It could be other organisms. It could be social insect behaviors, whatever. But there is this evolutionary aspect to it. There is this innate aspect to the behaviors. Sometimes they're different forms of learning. Sometimes they're different forms of behaviors. Like, for example, if you wanted to build a NeuroAI model of octopus cognition or octopus behavior, it would look very different from human cognition. And so you couldn't use the same kind of tasks to evaluate it, although octopi are very much like humans and that they're very Machiavellian. So building models that can pass the embodied Turing test will provide a roadmap for the next generation of AI. So that's the sort of their argument. And so go ahead.
Well, I just wanted to say, I love the octopus example,
Yeah.
and especially the Machiavelli reference. But the people have been doing this with dogs, and again, somewhat relates to the canine behavioral genetics project. Somewhat relates to some people at Emory who've been trying to scan dogs and use them as a model system. Super interesting, but in getting at how to, I'm sure everybody's familiar with Francois Chollet's is it ARC project? Like how to restructure an intelligence test to be more to extend it beyond the kind of gameplay tasks that have been used traditionally.
Yeah.
And the canine researchers hit the same problem in sense of like, how do we make intelligence tests for dogs?
Right.
But it's been really interesting to read in terms of what they have kind of discovered. And what I love is this finding particular dimensions in which dog cognition varies across dogs and breeds.
Right.
So it speaks to that in the sense of like, yeah, we need to, obviously we can't give them language tests,
right.
But that's the super basic one. But then it becomes, well, so how do you? And I think it speaks exactly to the kind of test differences or test tailoring that you're talking about. And yeah, really interesting literature that people can follow.
Yeah. So in this paper, in the first part here, they say historically neuroscience has been a critical driver and source of inspiration for improvements in AI. And so even if we're not interested in NeuroAI, we're still interested in what the brain is doing because that's sort of the basis for intelligence and like cognition in the brain. But then we have to think about what is it about intelligence that we're interested in, because there are a lot of brains in the tree of life. There are a lot of types of cognition in the tree of life, particularly those that have made AI more proficient in areas that humans and other animals excel at. So you can think about different domains like vision, reward-based learning, interacting with the physical world, and language, and of course language being nonverbal, as well as verbal or there are different codes, like, olfactory codes and other things that you can use as a stand-in for that. So there, you can make, it's hard to make analogies between like octopus language and dog language and human language, but you could, so you could characterize those things. It would be a little bit more compartmentalized, though, in terms of the tests that you would do, specific tests for the specific thing that your AI would specialize in. I mean, if it were a general intelligence, we've tried to master general intelligence, but especially at this viewpoint, it's even harder to see like what that would look like, because we've kind of thought of general intelligence as like the things that humans are good at, but maybe that's not the way to go. Maybe we need to think even broader. So they talk about von Neumann architectures and how that sort of contributed to our idea of, computation and things like that. Kind of thinking about like some of the things that the interfaces between computer science and neuroscience. So the Hubel and Wiesel work on visual processing has been very influential in terms of like pattern recognition in computer science. And of course, then that's influenced AI in our view of artificial general intelligence. So that there is this general intelligence that we can characterize is a thing that sort of is, for one thing, it's kind of grows out of the idea of universality, which is where, we think that, well, there obviously have to be these universal rules to intelligence, but also this idea that, we're sort of human-centric, and we know kind of the sort of nuts and bolts of intelligence. And if we can specialize in different types of intelligence, but we have to have a general intelligence to really have like this flexibility. Now, when we think about general intelligence, of course, we're thinking about humans, but of course, when we go beyond humans, and actually quite a bit beyond humans, then we begin to see that that's much different. That view of general intelligence looks much different. So, so this kind of goes through a lot of this. This points out the Turing test versus the embodied Turing test. So this is where we have a comparison between the performance of AI systems and their living counterparts. So in the Turing test case, we have a computer versus a human, but it's not embodied. On the left, the original Turing test is proposed by Alan Turing. If a human tester cannot determine whether their interlocutor is an AI system or another human, so you have a human talking with a computer, maybe it's a large language model that's generating, or a chatbot that's generating text, and it's answering the human's questions, the human's asking questions, the chatbot is giving an answer, and the human has to determine whether that's a human or whether that's an AI. And so that's the idea. And so if a human tester cannot determine whether their interlocutor is an AI system or another human, the AI passes the test. So that's the way they view the Turing test in the sense of AI systems, and how you can say whether it's like a human or if it resembles human or not. On the right, we have the embodied Turing test. So the embodied Turing test is where you have an animal model, which is this robotic beaver, and then you have this real beaver. And so they're going to be competing or comparing notes. So this is where this is embodied in an organism or a model that is like a robot or something like that. So an AI animal model, whether robotic or in simulation, passes the test if its behavior is indistinguishable from that of its living counterpart. And so, like I said, you can look at a C. elegans under a microscope, and it can do these omega turns, and it has a behavioral repertoire. And then, you could look at a simulation of C. elegans where it's doing the same omega turn, and you can say, okay, if that's indistinguishable, those two things are indistinguishable, then you say they passed the embodied Turing test. And it says no AI systems are close to passing this test, which is likely true. Here, an artificial beaver is tested on the species-specific behavior dam construction. So here you have dam construction, where the beaver has to assemble a dam, and you would have a robotic beaver, and a robotic beaver I don't think has ever come close to that. Now I give the example of C. elegans because I wanted to point out that actually you could simulate C. elegans behavioral repertoires quite easily compared to a beaver. And so you could simulate these behaviors using like a physics model. So the underlying model has nothing to do with the C. elegans connectome. You could still simulate behaviors. I mean, we can do this with like biophysical models because if the behavioral repertoire is simple enough, if it doesn't involve a lot of complex planning or whatever, then it would be quite easy to simulate if it's based on largely in physics. But you still wouldn't have that NeuroAI aspect of it. So that's one issue with this embodied Turing test that I can see that you depending on the behavioral repertoire that you're looking at, you could simulate it without having like a brain behind it. I mean, you could have maybe a robotic beaver build a dam without having a brain. It would just kind of like follow commands and but I think you have to unpack this embodied Turing test a little bit to see where we're going with this. So they propose an expanded embodied Turing test. This is one that includes advanced sensory motor abilities. The spirit of the original Turing test was to establish a simple qualitative standard against which our progress towards building artificially intelligent machines can be judged. That's this one on the left. This embodied Turing test would benchmark and compare the interactions with the world of artificial systems versus humans and other animals. Similar ideas have been proposed previously in 28 through 32. However, in light of recent advances enabling large-scale behavioral neural measurements, as well as large-scale simulations of embodied agents in silico, we believe the time is ripe to instantiate a major effort in this direction. So this is a nice call for like, okay, we have the technology, we have the ability to kind of look at this a little bit more in a sophisticated way than say they did in the 50s and 60s. Time to do it. Now they say that as each animal has its own unique set of abilities, each animal defines its own embodied Turing test. So depending on your model organism, it's going to look different. So this 28 to 32, these references, let's go down to the references. This is Rodney Brooks, Intelligence Without Representation. This is a classic paper from 1991. We talked about this paper, I think, recently in the group. Meyer and Wilson, From Animals to Animats. So this is a conference from animals to animats, and there are a number of different proceedings. This one being from 1991, which suggests to someone who doesn't know about that area that or maybe that knows about that area, this has been going on a long time. People have been using animal inspiration and biological inspiration for a long time. And there's a lot of literature that's out there that's decades old, but it may be useful today, that have kind of gone under the radar. And so that's what they're trying to bring that above the radar. This paper, Pfeiffer and Shearer, Understanding Intelligence, that's a 2001 paper. I don't know why they didn't cite the whole paper, but here's the DOI link. Pfeiffer and Bongard, How the Body Shapes the Way We Think, A New View of Intelligence. That's a good book. Josh Bongard and Rolf Pfeiffer, they talk about embodiment there. That's a good book. Good citation. And then number 32, Why We Need a Physically Embodied Turing Test and What It Might Look Like. This is from AI Magazine. And this talks about this idea of a physically embodied or an embodied Turing test. So it's very much an open question as to what this should actually look like. But I definitely say that, we actually have done a lot of stuff in embodiment. And that's something maybe we could, contribute to in some way. So this NeuroAI Grand Challenge, this is, AI has a number of grand challenges where people work on problems to try to gain insight and maybe solve problems at the same time. This is something, this embodied Turing test, this is the thing that they're kind of the core of what they're advocating. It's imitation. It's based after Alan Turing's idea of the imitation game. So this is something that, and we have to think about what animals do in terms of like overall, computing efficiently, engaging their environments, behaving flexibly, and so that's.
They should have cited what's the it's like the 1994 intelligent behavior in robots and animals, like a dynamical systems approach. It's the woman is it like Esther Thalen?
Oh yeah, Thielen and Smith.
Yeah. Sorry, that like seemed to be very much in that. I mean, not necessarily the embodiment, but well, it was robots and animals though in that book.
Yeah, they were. Linda Smith, of course, does developmental stuff, and Thielen and Smith. Yeah, that was a good book on like this approach to like embodied intelligence, but developmental embodied intelligence. And so this is not a new area. This has been going on for a while. The question is like, how do we crack the nut on this, or how do we get to the like, how do we get to something that maybe is approaching some sort of general AI or very good specific AI. So this is, they lay out a roadmap in the paper and they kind of go through what we need. So we need a new generation of AI researchers who are cross-disciplinary. We need support from, on supporting fundamental theoretical and experimental research. We need a shared platform capable of developing and testing these virtual agents. And then we need funding. So these are all things that are very basic. I don't know, like there's no like, we need specific things for an embodied Turing test, but it's typical of these kind of papers where they give a sort of an overview and then they say like, well, we need more funding. So yeah, Amanda had to go. Thank you for attending. David Fowl, yeah, okay, that's who that is. And then Jesse had to go as well. And it looks like
David Fowl had the very disparaging remarks on the NeuroAI. Has done good work as well. But again, we all well, I don't know if we've all. He's not the first person who said something inappropriate on Twitter.
Oh yeah. Looks like Lukas joined us again. So welcome back, Lukas. Hope you enjoyed that last part. So yeah, that's it for today. I think that's a good stopping point. And again, I'll post that in Slack, and maybe we can talk about these things more going forward.
Yeah, I want to check it. I'll check the recording because I kind of missed the last like 5 to 10 minutes of what you were saying, but I'm glad you shared that paper. So thank you.
All right, thanks. All right. Well, if there's no other comments or questions, let's wrap it up for today. And
okay.
Yeah.
Take care, everybody.
See you next week. Take care. Thanks. Bye.
Take care.